We're back with another teen science. Okay. Oh, oops. It's okay. Hey, what's up, folks? I'm Divya, one of your cafe coordinators, and we're back with another Teen Science Cafe live stream. Now, before we start with the stream, be sure to take a second to go down, smash that subscribe button, like the video, and turn on bell notifications to make sure you never miss another jingly NCMNS video. Remember to be respectful of all the guests in the chat, as our moderators will be keeping an eye out for inappropriate comments. Also, check us out on Twitter, Facebook, and Instagram.
Tonight, we've got an exciting guest for you. Our guest is a neurochemist and a community-engaged scholar who works at NC State University. Her job includes an exciting combination of both neuroscience and chemistry. Please welcome the one and only, the lightweight champion of the world of neuroscience, dopamine dictator, serotonin savior, adrenal admin, the fight no flight warrior, Dr. Leslie Sombers.
Well, thank you for that introduction. I'm really excited to be here to tell you a little bit about the work that we do in my lab today at NC State University in the Department of Chemistry. So what do I do now? Just share the screen? Okay. Okay, so I'm very excited to tell you about some of the work that my really talented research team is doing at the university. We're comprised of graduate students, undergraduates, and postdoctoral researchers, and we are electrochemists who use our mad electrochemistry skills to measure molecules as they fluctuate in the brain in awake animals that are making decisions. And we do this to try and get a better handle on how certain neurotransmitter fluctuations underlie specific aspects of behavior. And in addition, how these systems, how we can interpret these systems when they go awry in the case of mental disease. Okay. So what I've got for you here is just a video, and it's not real neurons. This is just some artist's idea of what it would look like in the brain if this very complicated structure of neurons is firing in the brain. And the point here is to try and help you understand what the brain might look like as it's working. The brain relays information from cell to cell. So the basic building blocks of the brain are individual neurons. And information is transmitted from neuron to neuron in two ways. Electrically, the signal starts at the cell body and it travels down the axon by way of an action potential. And when it gets there, vesicles full of neurotransmitter are released. That chemical signal can diffuse across the synapse where it can work on the next cell. And so chemical transmission is both electrical and chemical in nature. And this is how information is transmitted in the brain so that basically it underlies the brain's response to every experience that we have. And so once this action potential reaches the terminal and the neurotransmitter is released, it's really the volume of this signal and the route that the signal takes through the brain that determines every aspect of what we perceive, think, feel, and do. Okay, so it's kind of hard to wrap your head around the brain, which is why I started with that artist's rendition. It's a little bit easier to think about the stars in the sky in the Milky Way galaxy, I think, and there are 100 to 200 billion stars in the Milky Way. Now, there are 86 billion, close to 100 billion neurons in the brain. So about the same number of stars that are in the Milky Way are the neurons in our brain, just packed into this really dense tissue. And even more complicated than that, each neuron synapses onto many different neurons. So with 100 billion neurons, we have about 100 trillion synapses in our brain, which makes things really complicated because each of these cells is going to release different chemical modulators and respond really in a very versatile fashion to all kinds of inputs. And so it's really understanding how the brain works in terms of electrical and chemical neurotransmission is really a huge analytical challenge. And that's exactly what we're working on in my lab at NC State University in the Department of Chemistry. So I have a question for you guys. Do you know what are some of the common neurotransmitters? I don't know how much neuroscience you get in high school, but I bet some of you know a few common neurotransmitters that we can list up here, right? Anybody care to say any? And while you're thinking about that, let me tell you a little bit more about this process of chemical neurotransmission. So when the action potential reaches the nerve terminal, what happens is these vesicles that are full of neurotransmitter are triggered to fuse with the presynaptic membrane. And when these vesicles fuse, they dump their chemical content into the extracellular space. This is called the synapse. Once it's in the synapse, the neurotransmitter can bind to a receptor on another cell. And different types of receptors do different things. They might excite the next cell, or it might inhibit the next cell, or it might just have a modulatory function, broadly speaking. And so, in this way, the signal is transmitted from this presynaptic terminal to the next neuron. And this signal is then propagated through the circuits of the brain to exact the functional output that we see as a behavior or a perception or a feeling or a response of some kind to some environmental cue. Okay, so some common neurotransmitters. I'm seeing adrenaline coming here. That's right, absolutely. Adrenaline is also called epinephrine. Dopamine. Dopamine's a very common neurotransmitter that people seem to be really fascinated with. It's really involved in motivated behavior and in movement. So we're going to talk a little bit more about dopamine today. But yeah, there are a bunch of neurotransmitters in the brain. Dopamine, serotonin, adenosine, epinephrine and norepinephrine, that's adrenaline and noradrenaline. GABA, that's a small molecule, yes, hydrogen peroxide. I bet a lot of people did not know that they have hydrogen peroxide in their brain, but they do. It's an important neuromodulator. Oxygen, glucose, lactate, neuropeptides. There are many different neurochemicals floating around in our brains doing really important jobs. And the idea here, what I'd like to find out and what we're working on is what are the jobs of, what do each of these neurotransmitters do, and how do they underlie the different aspects of our behavior? Okay, but all this, I started right into the neuroscience, and my official training is in electrochemistry. So you guys were really good at coming up with some great ideas on what are some neurotransmitters. Here's another question. Does anybody know what is electrochemistry? See if I can give you a clue. Well, while you're thinking about that, let me tell you a little bit about myself. So before I started this research lab at NC State using electrochemistry to measure molecules in the brain, I was once a young science enthusiast, just like you guys. And I went to high school in a ski town in northeastern Pennsylvania. I went to Pocono Mountain High School, where I learned a little bit about everything, and I learned a lot about skiing. And then I went to college at the College of William and Mary, where I became really interested in chemistry. and in particular in analytical chemistry, in measuring how much of different molecules are in a sample and what molecules are present. And I did a bunch of work as a master's student at William and Mary, analyzing levels of heavy metal in fish tissue, because William and Mary is there on the Chesapeake watershed area, and there's a lot of industrial runoff, and heavy metals and other nasty chemicals get into the water, and then fish eat that stuff, and then the people eat the fish, and this could be a health concern. So state governments typically make rules for how much of wild fish, a given type of wild fish, should be consumed in a given amount of time. And so as a graduate student at William and Mary, I worked for the Virginia Institute of Marine Sciences, and I analyzed fish tissue for various metals so that the state government could make appropriate recommendations for fish consumption. And then I decided to go to graduate school for my PhD, where I learned all about electrochemistry at Penn State. So, electrochemistry, what is it? Following electrons in chemical reactions is what I'm seeing in the chat. All right, yes, and do you know what else it is? It is the chemistry that happens inside of a battery. So it is the controlled flow of electrons that makes possible everything that you guys love. Phones, video games, computers, all of it runs on batteries, and all of that battery technology is electrochemistry, the controlled flow of electrons for powering devices. So when we talk about electrochemistry, we often use the term redox reaction. And a redox reaction is a chemical reaction in which one substance loses electrons, it's oxidized, and another gains electrons, that substance is reduced. And you might learn a little bit about this in your high school chemistry, I'm not really sure, but you'll definitely get it in college. It's a real important field because we're facing an energy crisis right now, and electrochemists are very important for discovering alternative or for making possible and developing alternative sources of energy that will help get us through this crisis. One thing that may help you out sometime in your high school classes or in your college classes is the term oil rig. Oil rig. I don't know if anybody's heard of that before, but it's a little saying that electrochemistry students use to help them remember that oxidation is the loss of an electron and reduction is gaining an electron, okay? But, batteries, they're really important, but they were never my thing. Even as a young child, I was really interested in how the brain works. And so when I went to graduate school, I was excited to learn that electrochemistry can be used for molecular measurements. And so that's what I pursued. Okay, I skipped one. So let me try and explain to you how we can use electrochemistry for molecular measurements, and even better, for molecular measurements in the brain. Electrochemistry is really amazing because we're making measurements in real time, basically live, just like this stream, while the electrode is implanted in the sample. And in this case, if we make measurements in the brain or at a living cell, the sample is this living organism that you can measure this molecules that are present in real time while the organism is going about its business and making decisions, living life, functioning as it normally would, so that you could then correlate the chemical measurements to the functional output or the behavior. So what we do is we make electrodes out of carbon fibers, individual carbon fibers, the same type of carbon fiber that's used to make your tennis racket or maybe a snowboard or a bike. It's a very lightweight and mechanically strong material. and it's also an excellent conductor. And so while these fibers are typically used in big bundles, we take just one carbon fiber that we pull out of the bundle, and each fiber is about 1/10 of the diameter of a single human hair. And we can insulate it and make an electrode out of it. And then this tiny electrode, which is much thinner than one of my hairs, what we can do is we can apply a potential to it. And this is essentially energy that will drive a redox reaction for an analyte of interest. So if, for instance, dopamine is in the vicinity of the electrode surface, we can force its oxidation, right? And when it's oxidized, it gives up 2 electrons to the electrode surface. So here now we have a redox reaction between the molecule, which is in solution in tissue, and the solid electrode. It's an electron transfer that's occurring between the solution and the electrode itself, which is different from all other types of chemistry in which both species that are reacting are typically in the same solution. Here we've got the electrode receiving the electrons is a solid, and the species that we're trying to analyze is a molecule in the solution. And that electron transfer can go the other way as well. It can be quantified, and we can use that information to inform us about how the system is working. Okay, so I learned all about this in graduate school at Penn State. And it was really a transformative experience for me, graduate school. It really, I learned all about electrochemistry, and it really pushed me to think about how I could use that knowledge to solve the biggest problems that I was interested in. And I was really interested in pushing the boundaries of how we understand information encoding in the brain and how that can be dysregulated by disease. I was frustrated by the lack of effective treatments for so many mental disorders. And I wanted to use electrochemistry to try and study the brain, because electrodes can be made really tiny so that they could be put in tissue and they can read out chemical signals essentially live in real time, rather than having to take material out of the sample and put it on some instrument and wait 10 minutes for the answer. You could essentially see what's there and how much of it while you have a probe in the awake animal's brain. And so this is what I decided to do. I learned how to make these tiny electrodes. In this case, I'm showing you an electrode that is, let me see if I can actually get a laser pointer. Ooh, I'm showing you an electrode that is much smaller than a single cell in culture. And I can put this electrode up against that cell, or I can even put it inside that cell. And if I apply a potential, I can detect the release of the neurotransmitter dopamine in this case. Or if I put the electrode inside the cell, I can tell how much is there at any moment. If I put the electrode up against the cell, I can stimulate this cell to respond by puffing on something that excites the cell. Usually a little potassium will do the trick. It's just a little bit of salt in solution. And this depolarizes the cell, just like a neuron is depolarized. And vesicles full of neurotransmitter will fuse underneath the electrode surface, and it can be quantified. Each vesicle that fuses dumps its molecules into this, essentially it's an artificial synapse that I've created here. And those dopamine molecules will then diffuse across that synapse, and they can be detected at my electrode surface. There are equations that can tell me exactly how many molecules are in each vesicle. And each of these spikes, so here's a recording from a cell, and it looks like this is maybe 3 minutes of data-ish. And this cell was stimulated where each of these arrows lies. And all of these spikes are individual vesicles that are fusing with the plasma membrane, dumping their content. we can blow up just one of these spikes, which is occurring on the millisecond time scale, and we can measure the current. In this case, it's picoamperes. Picoamperes, I don't know how well you know your metric prefixes, but if you don't know them, you're gonna need to know them if you're gonna do some science. Pico is 10 to the minus 12. So that's 12 orders of magnitude less than 0. Tiny, tiny amounts of current. But that current can tell me how many molecules are in each vesicle, and the length of time for each spike tells me how long it took for that vesicle to fuse. And so this is what I learned to do in graduate school. And I learned how to put different drugs on these cells to see how the drugs changed the time course of each individual exocytosis event. And it was really exciting. I was fascinated by neurochemical signaling, and I decided I wanted to learn more about it. And instead of just working at cells in a dish, I really wanted to see how this affected behavior in an intact organism. So I moved from Penn State and I went to UNC Chapel Hill to learn about the brain. And so, here's a diagram of some of the structures in the inner brain. And the purpose of this slide is simply to show you that the brain is not just one big organ. It's divided into these smaller little nuclei, which are in essence organs of their own. And they're all connected in circuitry that plays really important roles in doing the physiological functions necessary to keep us alive. So I went to Chapel Hill and I worked in a lab that combines electrochemistry and neuroscience. But instead of working in humans, I worked in lab rats. I'm having trouble getting my slide to advance right now. What's happening? Yes. Okay, so this is an image of a rat's brain, and it is also organized into separate nuclei that are complementary to the nuclei in the human brain. The rat brain is an excellent model system for the human brain, and we could use this to understand a little bit more about neurotransmission. So here's a side view drawing of a rat's brain, if you were looking at it from the side. So the back of the brain is here, and the rat's nose would be up here. And the whole thing is about the size of a walnut. And we're really interested in these dopaminergic projections that go from the ventral or lower part of the midbrain rostrally toward the front of the brain. And there are two main projections. The first one is the mesolimbic dopamine system, which is shown here in blue. It goes from the ventral tegmental area up to the nucleus accumbens, which is also referred to as the ventral striatum. And then there's a neighboring nucleus called the substantia nigra, which projects up to the dorsal striatum. And these two projections are involved in very different things. The mesolimbic dopamine system is involved in motivation and reward. And the nigrostriatal dopamine system is largely implicated in motor function, but there's a lot of overlap there, I would say. It's really not cut and dry. And so this is what I learned about at Chapel Hill. And I decided I would implant electrodes in these structures to try and learn more about what is happening there and how that relates to the animal's behavior. And the problem, the disease, the disorder that I was first interested in is addiction. And addiction is a disorder that is characterized by compulsive drug seeking and use despite harmful consequences. So the question that I have for you guys now is, do you think that addiction is a disease? Okay. I'm telling you that it is characterized by compulsive drug seeking despite harmful consequences. And it's a disease that affects a lot of people all over the world. And probably a lot of us have experienced or seen some of its repercussions, maybe in friends or family members, or certainly by looking at the news. And so I'm kind of wondering if you guys think that this is a disease, or how you feel about addiction. And while you're thinking about that, I can tell you that it is certainly a large societal problem. And it has many consequences: medical, social, economic, etc. And estimates of the financial costs of addiction are enormous. They can exceed $1 trillion annually. And these are old numbers from NIDA, at least 5 years old. So this is probably worse now. But so the consequences of drug abuse and addiction are vast. And we're trying to work on that a little more. But I'm really interested to hear what people are thinking about whether or not drug abuse is a disease. I'm seeing some people are saying that it's a disorder, but I haven't seen anybody say whether or not it's a disease. So I'm going to take that as a no. And unfortunately, a lot of people think that it's not a disease, but I'm going to try and convince you otherwise. Here I'm showing you some brain scans from an individual. At the top, these are 2 brain scans from an individual who, from a control individual and from a cocaine abuser, an individual who abuses drugs. All right. And so you can see there are, if you can see nothing else, you can see there is a big difference between these 2 images. Below is an image depicting metabolism in a heart disease patient. So the heart disease patient is on the right and the healthy heart is on the left. And again, you could see there are very big differences between the diseased heart and the control healthy heart. I'm having trouble making my slides. So I think this picture shows you that addiction is a lot like other diseases, such as heart disease. All right. It can change an organ's structure and function in very measurable ways and in very predictable ways. And those changes can be long-lasting and can lead to harmful behaviors and consequences in the people who abuse drugs. And so because the drugs change the brain structure and how it works, it can be classified as a disease because, for instance, in heart disease and in drug abuse, both of these disorders disrupt the normal, healthy functioning of the affected organ, have serious consequences and can last a lifetime. So for these reasons, I'm going to go ahead and convince you that addiction is a disease. That is a serious disease that has tremendous societal cost. And so I showed you some of the costs of addiction, the medical costs and the economic costs. But drug abuse and addiction is a problem that I can really relate to. And here's a picture of my friend, Jimmy Sweats. And this is the real cost of drug abuse and addiction. Jim was my friend in high school, just when I was in your shoes, right? This guy and I, we hung out and we were friends. This is a brilliant guy. He died at 39 years old, about 2 years ago. He died of an overdose. He was amazing. He was brilliant. He was good looking. He was fun to be with. Everyone enjoyed being around Jim, and everybody who met him thought he was amazing. And he graduated from our high school at the top of his class. It was a huge high school, 500 something in the graduating class. He went on to the University of Pittsburgh with tremendous potential. He did fantastic in college, had a scholarship to study ecology in Tanzania, went there for something like 3 months. And he was a nature guy, learned all about this other culture and learned how to speak Swahili. When he returned to the States, he worked as an environmental consultant for the Navy in DC to help figure out this tragic whale death situation where whales were dying. And he then went on to George Washington School of Law, where he again graduated at the top of his class. But along the way, something happened. And he was introduced to drugs that changed the way his brain worked and changed the way that he perceived his environment and changed the way that he prioritized his commitments and his changed the way he prioritized environmental inputs, I would say. And in the end, he died about 2 years ago. And it was really, really tragic. And it's this event that made me realize how important my work really is. I study the brain and I try to figure out dopamine release and the release of dopamine's friends, other neurotransmitters, to understand how and why this is happening. And this really hit home for me. It really made me understand why this is such a big problem, because this disease is taking beautiful, brilliant people who have so much potential. And what I felt and what I feel now is still just a drop in the bucket compared to what his family, his mom, his dad, his sister, and his brother feel. They miss him every single day, and they never imagined that this would happen to them. Addiction is a disease, and it is a problem that is vastly underfunded. And this is despite the fact that it is a huge public health problem, and way too many brilliant, amazing people have died due to this disease. And there has just been way too much loss. So you can put up slides all day showing you how much it costs your average American. But it's when you lose a friend, a loved one, a son, a brother, that's when you realize what the magnitude of this problem. And so that really pushed me to want to help more people who are suffering because they have a loved one that is abusing drugs and who feel helpless. And the best way to help them is to try and understand the molecular mechanisms that are driving their drug-seeking behavior. And so that's what we do. Okay, let's move to the next slide. I'm having trouble advancing my slides. I think it just takes me a minute. All right. So, drugs of abuse, they are different, all of them. There's all kinds: alcohol, nicotine, cocaine, opiates, other narcotics. There are, amphetamines. There are many different types of drugs of abuse, and they all work on different proteins and by different mechanisms. But one very interesting thing is that all drugs of abuse induce a big surge in extracellular dopamine. And I'm having trouble with my chat. There we go, I'm moving that over. Okay, so the way that this happens is, I told you about how these vesicles full of transmitter fuse with the presynaptic space and they dump their chemical content. So if a drug is on board, it's going to work on different proteins that are around this terminal. But in all cases, it's going to increase the amount of dopamine. So for instance, cocaine, right? So what cocaine does is it causes more dopamine to be released than normal. And on top of that, it blocks this dopamine transporter up here. This protein serves to suck dopamine out of the synaptic cleft and bring it back up into the presynaptic cell, kind of a recycling mechanism, so it can be used again. And if you turn off this recycling mechanism, the dopamine is going to stay around in the synapse longer, doing its job longer. And what this does is it causes a euphoria and a desire to repeat the experience. So when the dopamine binds to dopamine receptors on the next cells, it binds on two different types of cells. The first is a D1 receptor containing medium spiny neuron. And what these are neurons that are thought to convert urges into action. So it binds on these cells that are important for having you convert your urge into an action. And it suppresses the activity of cells in the opposing circuit, the circuit that is engaged when you basically weigh the consequences of your actions and you think, should I do this or not? You know, what's really going to happen? Well, the brake system is dampened all at the same time. Eventually, repeated drug use leads to long-lasting changes in the neuron structure and function that underlie all kinds of aspects of drug abuse, tolerance, withdrawal, and other persistent consequences. Repeated exposures trigger the release of more and more increasing amounts of dopamine that then diffuse further and further, potentially ratcheting up that urge to use again. It can ultimately decrease the number of dopamine receptors there, which alleviates the overstimulation of the dopamine system problem, but then it can exacerbate withdrawal. And when you feel horrible because you're in withdrawal, you're searching for some kind of medicine to make you feel better again. And it can really compromise the user's response to natural rewards, like food or taking care of the children, things that are naturally rewarding. And it could also limit the brain's ability to respond to new experiences. And it's really the brain's response to experiences that is learning. And so what this does is it limits a user's ability to develop new behavioral alternatives to drug taking. So it's a very complicated thing, but I think the important thing is that all drugs of abuse cause this big surge in extracellular dopamine that is very short lasting, milliseconds, but it happens repeatedly. Okay. Okay. So another thing that we know is that environmental cues are important. These cues are motivational stimuli or things that we see in the environment. You know, maybe it's the Starbucks sign outside of the Starbucks that really makes you want to go in and get that coffee. Or maybe it's, maybe you just love, I think Starbucks is the best example. I mean, my teenage daughter just loves Starbucks and she doesn't really want it until we're driving by the Starbucks and she sees it and then she really wants to go in, right? And what she's seeing is a cue. It's something in the environment that is activating her dopamine system and causing dopamine release in a part of the brain called the ventral striatum. And so what is it that changes such that this dollar bill, which used to signal, oh, I can buy food for my family and this will be naturally rewarding to me, what makes it, for some people, this cue then becomes an representative of when they see that $100 bill, they think, oh, I need to go get some cocaine. Right. And so what's happening here is that over time, dopamine response to natural rewards becomes compromised, and you the brain responds to these cues that are paired with previous drug use and availability. And it's exactly that for this reason that you see things like stars have drug problems and they go into rehab and they tend to recover and they're good in that very controlled environment. But when they get out and they see their friends with whom they used to do drugs, or the place where they used to buy drugs, or something else that is paired with that drug, they feel this irresistible urge to seek drugs at the expense of their other consequences, or of their other responsibilities. Okay, so if all drugs of abuse cause this big surge in dopamine, what we want to know is how that is happening and when that is happening. Because I think that's the key to developing effective therapeutic interventions, whether they be pharmacological interventions or behavioral interventions. We really should know when the dopamine is being released if we want to try and control that, if the dopamine is the driver of the behavior. And it may only be, it's almost certainly only a partial driver of behavior. There are many other neurochemicals also involved, but it's very striking that all drugs of abuse cause this surge in dopamine. And so what we do is we can implant one of our little electrodes into the nucleus accumbens of an animal and measure that dopamine in real time in the volitional process of drug self-administration as they are deciding whether or not to take a drug. Because at Chapel Hill, I learned how to do these surgeries, and I learned all about animal models of decision-making as it relates to reward consumption. And so I'm going to show you an experiment that we did where we implanted electrodes into the accumbens, and we put the animal in a controlled environment, a closed box, where we could control lights and sounds, and have a lever come out so that the animal could self-administer a reward, in this case, a drug of abuse. And so the animal is deciding this. And what we did was we did something like a Pavlovian conditioning experiment, kind of like Pavlov's dog. I don't know if you've ever heard of this in like a psychology class. But Pavlov rang a bell before he gave his dog a treat. And eventually the dog would salivate just when he heard the bell, because he knew that the bell meant a treat was coming. Right? So we do the same thing with the rats. We turn on a tone and a light 3 seconds before a lever comes out, and they can press that lever to give themselves an infusion of drug. And so we can see what's happening as they see the environmental cue, and as they walk up to the lever, as they press the lever, and as the drug hits the system. Okay, so I've told you that all drugs of abuse cause this dopamine surge in the nucleus accumbens. And I also want to tell you that this video that you're about to see does have an animal with electrodes in his head. So if you are not comfortable with that, feel free to just turn off the video for a few minutes. But what's happening here is I want to know what you think is going to happen. So in this experiment, a light is going to change. It's over here on the left side of the box. And 3 seconds later, a lever is going to come out over here, and the animal can press it to get a reward, a non-natural reward. Now, what do you think is going to happen? When am I going to see the dopamine signal? Anyone in the audience have a prediction? They're going to see the light, and this animal is going to turn and it's going to walk over and press the lever. So at the beginning of this experiment, when they don't know what the light is, what do you think is going to happen? When will I see dopamine? Does anybody have any idea? Does anybody even want to guess? Come on, guys. This is, you know, it's so much easier if it's a conversation. Waiting for responses. But, you know, so the animal, he's in the box and he doesn't quite know what everything is, but he'll accidentally bump the lever after the light comes on, and wondering what you think is going to happen. I can tell you that when he presses the lever and he gets that reward, there will be a surge of dopamine, that's for sure. And then things are going to change. So after many presses, let's say 50 to 200 presses for this rat, he's going to learn that the light means drug will be available soon. And just like the salivation in the dog shifted from when he got the reward to when he heard the tone, the dopamine response is going to occur with the light. And so here's how that looks. So I have to move some things around to get to the play button. Oh no, maybe it's a laser pointer problem. Okay, all right, so the animal's in the box, lighting changes, and then he presses the lever. Okay, so this animal is now well trained. When he first started pressing, we got a dopamine response only when he pressed the lever, when he was, retrieving that reward. But over time, as he learns what that light and that tone mean, when the light and the tone come on, we see a big dopamine response in anticipation of reward availability. And when he presses the lever, there's more dopamine that's released. So what's happening here is this cue that indicates reward availability is now taking on more meaning, and it is in and of itself evoking dopamine release. And that dopamine is acting on certain neurons that convert urges into action, and he immediately goes over and presses the lever. So I'll play it for you again. You can see right when the house light changes, his dopamine levels around the electrode go way up and come back down. He immediately shifts his attention to the lever when that dopamine goes up. And he goes over and presses, and then we get the dopamine response in response to the primary reward. Okay. So what this tells us is that this cue evoked dopamine can drive a very powerful urge to consume drugs. And what we can then do is we can use some tools, chemical and genetic tools, to try and dampen that signal or control it in some way to turn those dopamine cells off and on in a predictable fashion. And we can then change the way they are behaving, change the likelihood that they'll press the lever or the time it takes them to go over and press the lever, which is a good indicator of their motivational state. Okay, so let me move on. Okay, so this whole first part of this talk was all about the role of dopamine transience in addiction. Dopamine does more than just motivation, though. The very next door nucleus in the brain also has dopamine neurons that project to the striatum. But these cells are different, and they are very involved in motor control. And in Parkinson's disease, which affects some 10 million people worldwide, the dopamine cells in this particular projection die off, whereas those in the mesolimbic reward projection remain intact. And so the question is, what is this dopamine in the motor loop doing? What is the dopamine in the nigrostriatal pathway doing in terms of action initiation and motor control? Now, Parkinson's disease is another big problem. I don't know if any of you know someone with Parkinson's disease, but they tend to be very stiff and slow. They have trouble initiating movement. They tend to have resting tremor. And so they're called, so they're essentially like frozen. It is a kind of a verbiage that people often use. And they're treated, the standard treatment for Parkinson's disease since I think the 70s has been treatment with L-DOPA. L-DOPA is the chemical precursor to dopamine. So all the dopamine cells die. In fact, 80 to 90% of them are gone before you get your first symptom, which is absolutely amazing. So you're completely symptom-free and you've lost almost all of your dopamine cells, which tells you how good the rest of the cells are at picking up the slack for those that have died off, right? When you get your first symptom, you've got almost no dopamine cells left in that part of the brain. And the other thing that we know, so if you treat the patient with the precursor to dopamine and load up the remaining dopamine neurons with extra dopamine, you can alleviate some of the motor problems and the patient can move again. Another thing that we know is that oxidative stress contributes to the initiation and progression of this disease. Now, perhaps one of the best known poster people for Parkinson's disease is Michael J. Fox. So he was a huge actor when I was young, when I was about your age, and he starred in this movie, Back to the Future, and everyone knew about Michael J. Fox. I mean, he was just a household name. And so he got Parkinson's disease in his early 20s, which is about when it typically strikes. It's most common in men. So he was fine up until he was about 20. He got Parkinson's disease and he was treated with L-DOPA. And with L-DOPA, what happens is you can move again, but over time, typically maybe 5 years, a large percentage of patients then develop these hyperkinetic, uncontrolled movements. So when they have the L-DOPA, they can move, but they also have all kinds of movements that they cannot control. So if you see Michael J. Fox in an interview today, you see he doesn't sit still. He can't really control his core and his torso. He can talk to you and he can initiate movement, but he can't control it in the same way that he could before he was Parkinsonian. And so we want to understand what L-DOPA is doing and why we have this progressive change over 5 to 10 years. Why does it work fine at first? And then these side effects, which are this movement problem is a side effect of the treatment, not the disease itself. And I'm convinced that if we understand the chemistry a little bit better, the neurochemistry, we could do a better job of treating it so that we don't have all of these side effects. Okay, so in order to do this, because Parkinson's disease also involves oxidative stress, we decided to use electrochemistry to monitor hydrogen peroxide and dopamine at the same time during dyskinetic behaviors in Parkinsonian, in a model, a Parkinson's disease model in rats. And so these animals have the dopamine neurons dead on one side of their brain. So one side of their body has trouble with movement and the other is relatively unaffected. And just like you saw in that video of Michael J. Fox, when you give these animals L-DOPA, they have their own dyskinetic behavior. They twist, they move their arms about, and because one side of the body works well and the other doesn't, when they start to circle and circle pretty robustly. Okay, so I'm going to show you what happens when we give a Parkinsonian animal L-DOPA. About an hour after the L-DOPA is administered, you'll see what happens here. And again, if you're uncomfortable with looking at the live animal video, now's a good time to tune out for just a minute or two. And so here we have the animal in the box. And this time, we're not asking him to do any kind of a task. I'm simply monitoring his behavior, and we're monitoring hydrogen peroxide in red and dopamine in blue. And we do this for this animal. We measured him for about 6 months, so 3 times a week for 6 months, to see how these signals were changing over time. And I'll show you what happens here. So when the animal starts rotating, and he can't, control that, we see these chemicals changing. So every time the animal rotates, I asked my students to color the data with a gray bar. So I'll play it for you again. You'll see that when he's rotating, what happens is the hydrogen peroxide signal goes up and the dopamine signal goes down. When he stops, they reverse. And this is very reproducible. There's always a surge in dopamine before the rotation. When the rotation begins, the dopamine is dropping and the peroxide is coming up. So this, we think, is a real important clue as to how to develop a better therapy for treating the disease. And so, the question that we had was, why is this happening and how can we stop it? And so we sat and we thought about it for a minute. And I said, well, if these dopamine cells are dead, because this is a Parkinsonian rat model and we've killed almost all of them on that side of the brain, who is releasing that dopamine? What cells are picking up the slack? And so we targeted serotonin terminals in that area. And we administered the animal a standard dose of an antidepressant, an SSRI, that targets the serotonin machinery, because serotonin proteins can pick up dopamine and package it and transport it, not quite as efficiently as it can do serotonin, but it can do dopamine because serotonin is structurally similar to dopamine. And so I thought, well, serotonin neurons, they might have what it takes to release dopamine. And we know that these fluctuations have substantial effects. So if serotonin neurons are releasing the dopamine, they don't have the necessary brakes in place to prevent large pulsatile fluctuations in extracellular dopamine. So let's put an SSRI on board and stop the serotonin cells from taking up dopamine, because that's what an SSRI will do. It'll prevent serotonin transporters from taking serotonin, or in this case, dopamine, from the extracellular space and packaging it up for release. And so what you're going to see is another video of that same animal here on the left, doing the same thing he was doing last time. And the chemistry is shown here in color. And I haven't explained these types of graphs to you at all, but the brighter colors means that a lot is happening. There's neurotransmitter being dumped into the area by the electrode surface. And this is a 5-minute recording. So you could see on the left, when I play the video here, this animal, he's got atypical behavior. He's flailing his arm. This is not how rats typically act. He's going to start rotating. And if you look at the chemistry, what this tells me is that there's a lot of hydrogen peroxide and dopamine being dumped into that extracellular space as he's doing this behavior. Now, this is the same animal over here on the right. And the video is playing. He's just not doing much. He's acting like a normal rat. He's just kind of chilling in the box. It's the same animal the very next day. We gave him the same dose of L-Dopa. We just put the SSRI, the antidepressant, on board first. And now you can see the chemistry is largely normalized. You're not seeing these big bright splotches of color, which are indicators of the current. So we aren't seeing huge fluctuations in those two chemical species in the accumbens, or actually we're in the dorsal striatum here. And you see his behavior is normalized. So this, I think, shows kind of the way that the findings can be translated into better therapies. Now, this is just in a rat, and so there would be many more steps before it was ever put in a human. But this gives clinicians the information they need to start to think about those questions and how to better treat their patients. So guys, that was a long presentation. I just want you to know that electrochemistry has so much potential for solving our biggest problems, not just the energy problem. We can use electrochemistry to monitor molecules to study things like drug abuse and Parkinson's disease, and to get a really good foundation of information that's needed to develop better therapies to treat these diseases. Electrochemistry has so much potential for so much more than energy. And this is my research team at NC State, past and present. We all came together for a big Zoom meeting recently. All of these guys contributed to the work in some way, shape, or form. A lot of undergraduates here. And they're an amazing team. They've done a fantastic job. The work is funded by NIH, NSF, Pine Research and Pinnacle Technologies are big contributors to our work, not financially per se, but they are collaborators with us. And it's funded by NIH and NSF. And I'm happy to take questions.
All right. Thank you, Dr. Sombers. So as Dr. Sombers said, our floor is now open to any questions you guys have. So please put them in the YouTube chat or add us on any of our social media accounts. And we'll try to get to as many questions as we can. So first was, why do you use rats compared to other animals in your lab?
That's a good question. Well, we want to use rodents. Rodents are great models because they're small and easy to maintain. I think mice are a great animal model because they can be genetically manipulated and you can really study genes that way. But mice are also really tiny. And so if it's got to carry electronics on its head, like usually my animals have at least 3 electrodes. It's easier to use a rat because they're about 10 times the size of a mouse and strong enough to carry the electrodes and stuff. A lot of work is done in primates, but they're very, very valuable animals. And so if you can do the work in a rodent, it's really a better system because they're not as valuable. They don't live as long. They don't take up as much space. And their brains are a very good model for how the human brain works. They reproduce quickly.
Yeah, that makes sense. All right, next we've got, why are some substances like caffeine called drugs of abuse and are labeled as bad, but other addictive chemicals that are considered good or accepted?
Well, that's another good question. So remember, addiction is clinically defined as the compulsive drug seeking and taking despite other harmful consequences. So if you continue to do cocaine and you don't show up for your job or take care of your children, well, that's clearly an addiction because you can't say no and there are very harmful consequences. But a cup of coffee is not necessarily the same. There are not as obvious harmful consequences in response to drinking caffeine or, drinking alcohol in moderation. If you can just say no, you are not addicted. But the thing is that many of these substances are very addicting, and different people have different susceptibilities to that. There's a lot of variability there.
All right, on a slightly different note, what advice do you have for high school students or maybe undergrad students who are interested in pursuing neuroscience as a major or field of research? And would you say something like medical school would be the best way of pursuing this?
Well, that's a great question. So, I was interested in the brain as a teenager, and I really wanted to go to medical school. And I thought for sure I was going to medical school. Anybody who asked me, I was going to medical school. And I went to college and I took all those pre-med classes and I did all of the volunteering that you had to do. And I took the MCATs like 3 times or something horrible like that. And I was convinced I wanted to go to medical school. And this is a huge hurdle. I paid thousands and thousands of dollars in medical school applications because that's what I wanted to do. I was going to medical school for sure. And at first I was going to major in biology, but I got really interested in chemistry when I had a great chemistry professor. And you could study chemistry and still go to medical school and study neuroscience, no problem. And so that's what I did. And then when I was doing my volunteer work, you have to work with doctors. And the more doctors that I talked to, many of them were very frustrated with the state of medicine at that time. This was in the late 90s. And it was at that time when insurance companies were becoming more and more involved in patient care. And essentially, many doctors felt that the insurance companies were dictating how they cared for their patients, and they were not happy about that. And the doctors that I talked to who treated mental disorders were frustrated by the lack of fundamental knowledge that was governing the therapeutic strategies that they were using. They felt basically that they were choosing the drug from a list of possible drugs and more or less kind of just guessing at which one would work and guessing at a dose and then titrating it up or down from there. And I sensed this frustration on the lack of foundational knowledge. And so when I learned, I decided that I would go to graduate school, and I went to Penn State, and I learned that electrochemistry could be used to study neurotransmitters in an intact system. I decided that that was a much better way for me to pursue my goal of trying to figure out how the brain works and trying to help people. That just seemed like a better choice for me. So it was really kind of surprising to me that I thought that I would go to medical school, but in the end, and I mean, when I say I thought, like I was 100% sure. And in the end, that is not what I did at all. So I would recommend pursuing your interests, because you'd be surprised. You could easily major in chemistry and study neuroscience. And in fact, then you would have all kinds of possibilities. So always keep your eyes open, be prepared for anything, because you never know what opportunities are going to present themselves and what you will find interesting in the future. And chemistry is awesome.
Yeah, thank you. So a more specific question regarding addiction, do you study what happens to dopamine levels in rats that are conditioned to receive the drug reward, but then they don't receive it? And will this give you information about withdrawal?
So that's another great question. So what we know is that if an animal is conditioned to receive a drug reward, but does not receive it, this is called reward prediction error. And what happens is there's essentially a pause in the dopamine firing. So typically when the animal receives the reward, the dopamine cells fire faster in like a burst. If the reward is omitted, there's a pause in the dopamine cell firing. And this is how the learning, this is thought to be how learning about cue reward associations is encoded. It's a reward prediction error. So you can measure the dopamine cell firing in this type of an experiment, or the dopamine release, and kind of learn about the learning process itself. I forget exactly how the question was worded, but I hope I answered it.
Yeah, I think you covered it.
Okay.
We've got, where do you see your research going in the future? And also, how do you see your field changing in the future?
Well, I focused this talk on dopamine because it's a popular subject, and people think dopamine is all about euphoria. But, really, dopamine is less about euphoria and more about the urge to do something. I imagine that if a whole bunch of dopamine was released in my ventral striatum, it would not feel good. I would feel very motivated to do something, and that to me would be unpleasant. And so I think the field is now changing from this dopamine-centric idea that dopamine is the answer to the idea that other neurotransmitters that are more difficult to measure are also playing a very important role. And I didn't talk anything about that in this talk. But what we're doing is we are adapting our technologies to the detection of other molecules that we think are really important in signaling the reward cue, the cue reward association, and in driving drug seeking or movement, right? So there's a motivational aspect, and then there's also the action initiation aspect. And in both cases, dopamine is not the only player. So we are trying to adapt our technologies to detect other important classes of molecules, especially endogenous opioids. So people don't really realize that we have opioids in our brain that do important work, but they're very difficult to detect. So now we're trying to detect the endogenous opioids with this technology and to pair that information with the dopamine information so that we can get a better picture. Right now, I feel like we're at the symphony and we're listening to like only the trumpets. And the whole rest of the orchestra is silent. And what I want to do is modify the technology so that we can now hear all the chemical players, or at least more than just the trumpets. And so we are working on the endogenous opioids, especially a molecule called metenkephalin. It's one of my favorite molecules right now.
All right, so unfortunately, I think we're out of time for the Q&A. I'd like to thank you, Dr. Sombers, for answering the questions, and I'd like to thank everyone in the audience for posing them. And now I'll hand it over to Alina for closing.
Thank you again to Dr. Sombers, and thank you all for watching. I hope you were shocked by everything you learned today. We would also like to thank the museum's digital media team for helping us put on the cafe. Don't forget to follow Teen Science Cafe and mention us on Twitter, Facebook, and tag us on Instagram. Also, keep an eye out for a follow-up email containing our survey, or go to the link tinyurl.com/teenscisurvey2 or scan the QR code that we will put on the screen shortly. Once you complete the survey, you will receive a ka-ching discount code for the museum store. Be sure to register and tune in for next month's cafe on Friday, January 15th with Dr. Casey Bush to learn about why we think what we think about climate change.
Thanks for joining tonight's Teen Science Cafe. Those of you who have tuned in to our previous cafes this year know that I'm the outro guy and that I have one very simple request. That you go down right now, like Divya told you to at the beginning, smash that subscribe button, electrocute the like button, and destroy the bell notification button to make sure you don't miss some other premium content from the North Carolina Museum of Natural Science. Don't forget about filling out the short survey after the cafe. My name is Sashank, and these have been your cafe coordinators. Stay safe this December, excuse me. Have a holly jolly holidays, and hope and pray, knock on wood, do whatever you gotta do to ensure that this year's energy does not carry on to 2021, because I don't think I can handle that. Ho, ho, ho. I hope you guys like this outro. Thank you.